for joining. Today's a very special day. Today's International Women's Day, and I want to congratulate all the women who are involved in potato agriculture around the world who are joining us today. Thank you for being part of this effort. And our chief leader here is Nora Olson, also a wonderful lady from Idaho. So Nora, thank you too for being one of the great potato people of the world. So I just want to share with you today from my office here in Canada. It's winter, and some of you probably are in the tropics where it's warm. And anyway, wherever you are, good day, and let's start our sharing time together. And I have to figure out how to start this thing. So my topic for today is about geographic constraints in potato breeding. And we're going to talk about the effect of day length and light intensity specifically as we look at how this affects our work of potato breeding and selection work and how our varieties perform in the field. And I have done this work for the last almost 40 years, or more than 40 years, in different parts of the world. So that's sort of why I feel this is important, especially for young scientists, young extension workers, that we understand the potato better and understand how the potato responds to day length and light intensity. So the first point I want to just share with you is about the potato plant. The potato plant has basically four components: foliar growth, or the haulms. It has roots. Then it produces stolons, which are actually underground stems. And we want to produce tubers, which is our goal. So we are going to try to understand how the partitioning of dry matter takes place, where it's produced in the leaves, why it first produces more leaves, produces more roots, then it starts to produce stems or stolons, and finally it starts to produce tubers. And what triggers these aspects of the different growth phases of the potato plant? And we know that the genotype plays a very big role in this, what variety, and also day length or photoperiod and light intensity. Here's a picture of two small plants. And you see the plant on your right has got very short stolons, and the bottom leaves are yellowing, and it's producing tubers now at a very early stage in its development. The plant on the left, you see the one very long stolon, which is very interesting. So this variety is actually Cooperation 88 from China. And you see how it's developing differently with longer stolons, and in fact, one very long stolon. So you can tell right away that this one is partitioning more slowly to the tubers, and that the signal for tuber production was much weaker because that's why the stolons are so long. This is fundamental because we want to grow potatoes as tubers to eat, and that's our goal. Looking at the world and where the potato comes from, the potato comes from just south of the equator in the Andean mountains of South America. The circle on the map shows you clearly where the potato originates, and this is where we have the greatest diversity of germplasm, but basically growing at around a 12-hour day length. From here, the potato has spread all over the world over the last 400 years. And particularly from here, it went first, of course, to Spain and Italy. From there, it moved in through Europe, as well as then from there, it traveled to other parts of the globe. It took over 100 years for the potato to be adapted to European long day growing conditions. If you go to the center of diversity in the Andean highlands of South America, you see a very huge array of different varieties. I think there's over 4,000 native varieties grown in the Andean region today. And this is a market in Cusco, and you see here, look at the potatoes here, and look at the diversity of shapes and colors. It's such a rich diversity we have of this germplasm. And my question is, and the challenge is, what's the value of this germplasm and how do we utilize it? That's where the International Potato Center comes into play. The center was established in 1971 in Lima, Peru, and its goal was to collect and distribute the Andean germplasm from the Andean mountain regions. First of all, collect it, describe it, and keep it both in situ, both up in Huancayo and different parts of the Andean region, as well as in vitro, as well as true seed. So this collection has been maintained by SIP very carefully, and this is available for scientists and countries around the world for use in their own breeding programs and for selection work. I worked at the Potato Center back from 1979. I worked for them for many years, and then later on I was on the board of trustees. So I'm familiar with how this center works and what its mandate is and how it has helped many of you who are probably listening today in the work of potato breeding work. I want to share with you just a simple example of how we used Peruvian germplasm at a location in Vietnam, which is at 12 degrees north latitude. And from there, how we've used that germplasm in northern parts of, further north in China, as well as in Canada, just to illustrate the effect of day length and light intensity on how germplasm from SIP and even from Vietnam respond to the different photoperiods in China as well as in Canada. So here we have Dr. Tung with his wife doing a crossing work here in Dalat at 12 degrees north, using a lot of locally adapted varieties crossed with germplasm from SIP for, in this case, primarily multi-mold genes for late blight resistance and some virus resistance and some heat tolerance. So this germplasm from SIP is exotic in a sense, but it's not adapted to the day length of even Vietnam. But so Tung and his wife are crossing this work and trying to produce progenies with improved traits that would be adapted to growing conditions in different parts of the Northern Hemisphere. This work has been funded primarily by the Unanoma University and also somewhat by Sunrise Potato here in Canada. So from those crosses, we have selected progenies, and some of these have come to Canada. And here in Ontario, where I live now, it's 46 degrees north latitude. So in the summertime, we have day length of 16 hours. And this is just a picture of 4 plants from true seed of one cross that are growing in pots here in Ontario. And you'll see a very interesting contrast. You have 3 plants with very long stolons, although one plant has produced tubers. You have the 4th plant, the far right, has no stolons, very short stolons, and has tuberized. So the selection rate for adaptation actually is quite low. And this in many ways is discouraging. You say, well, what's the use of doing these kind of crosses because the chances of success are so low? Well, even though the odds are low, the genetic makeup is so important that we don't want to forego that. And therefore, I say this is important that we still utilize such germplasm from Peru to make improved late blight resistant chipping varieties, in my case in Canada, for our changing environments here in these latitudes. And just to look at it more carefully, this is another clone that's growing here in Ontario, Canada. And you see this is a plant that's growing in mid-season, and you see rather long stolons. And in fact, you see some plants that even have branching on the stolons, which is not really what you want. And this will be a late maturing clone, but as the fall season comes along, it'll be able to tuberize better. But likely this clone will be rejected because the stolon length simply is too long, so the adaptation to long days just isn't quite there. So from this material we've gotten from Vietnam, I have, this is a picture taken last year. We had about 3,000 clones for 2nd generation that we were growing out in the field. So you see they're flowering profusely, large canopy. This is probably taken after about 6 weeks after emergence. And from here, we were able to select less than 5% of the clones were able to select for adequate adaptation to long days in our conditions. I consider that still quite good because even with that lower level of selection, we still got 40-some clones that we thought were okay for further evaluation in our selection work. Now, take some of this same material from Tung, from Dalat, which is at 12 degrees latitude, to parts of Yunnan province in China. Here is in Dali, and here we're at 27 degrees north latitude. And you see that a selection from Dali is growing, and it's probably 2 meters tall. Now, this plant is growing during a rainy season, so there's low light intensity, probably a little bit too much fertilizer. So this plant has shown severe delayed partitioning to the tubers and just keeps producing more foliage. And this is not what you want. So this clone actually, you would say, is poorly adapted to the longer days and low light intensity of Dali. However, if you would manipulate this through lower nitrogen levels, some growth regulators, boron application, you could probably make these plants shorter and partition earlier and be more successful. But understanding the behavior of this variety, then you have to look at it. Is this variety worth keeping? Does it do well under short days? And if it does so, then we should manipulate this through other ways to make it more productive. It has very good resistance to late blight, this variety, and it has very good processing quality for chips. Those are the pros. The cons are it's late maturing. And this is just to show you the contrast. Here, the picture on your right is a summer crop in, actually this is Shantung and northeast Yunnan province at 27, 28 degrees north latitude. Flowering profusely, beautiful crop, late maturing summer crop. That same seed goes to the lowlands in the winter season under short days of say 10 hours, 11 hours. There's no flowering, small canopy, and very quick senescence. Probably the plants mature in 70 to 80 days. And very nice adaptation to these kind of conditions. And just to show you another picture of Cooperation 88, which is an amazing variety, really. Here on the right, you see a picture of the potato growing in Yunnan. And here we're getting about 30 tons per hectare in 75 days. Beautiful crop, winter season crop. In contrast, on the left is a picture of the same variety grown here in Canada at 46 degrees north latitude. And you see after even a long growing season, the plants are still growing vegetatively. Partitioning is not taking place, and you have many very small little tubers. So clearly this variety is of no use under long day conditions. However, for genetic breeding purposes, this variety offers some very important traits which we want to utilize for late blight resistance. It's multigenic, for virus resistance, and even for processing quality. It has some very good qualities and eating qualities as well. So those traits we want to utilize, but it would be discouraging to think how this will work. But that's why I'm talking about this today, to encourage us not to give up, but to utilize material like this in our programs. Now, there are exceptions. Now, the variety Unica, which is a variety which was selected and grown in Peru with SIP's work and the Peruvian government's work, this variety has shown a uniqueness that it's widely adapted to many parts of the world. And this is a variety that's called Qingsu 9 in China, which was selected in northern China at probably 40 degrees north latitude. And it's growing in many parts of Asia as well as of Africa today. So this variety is showing a contrasting kind of behavior. So the genetic makeup makes this one less sensitive to photoperiod. And here's a picture taken here again in the Shantung area of northeast Yunnan province in the summer season under long days of low light intensity, and it's producing a very nice crop, not excessive foliage. So there are exceptions to how these varieties respond to day length. And to find varieties like this actually is very desirable because this gives you much more flexibility in where you can grow it. Now, if you go to East Africa is kind of unique because it's also an area that's close to the equator. So actually quite similar to what we have in the Andean highlands of Peru as far as latitudes go. And here the SIP in Andean germplasm has done very well. Has been very easy to grow this material in East Africa. In Rwanda, where I used to work, we had great success with SIP material from Peru that adapted well, and we were able to select good varieties for late blight resistance that have done very well and are grown throughout East Africa. And here's a picture from Ethiopia. And again, it shows that for that part of the world, the Andean germplasm is actually very easy to utilize and has been used successfully as well. So, just to summarize, moving genetic material from the equator to further north and south latitudes, you always have an issue of long days which delay maturity. Therefore, they're difficult to get them to tuberize well and to bulk early. Therefore, you'll have very low selection rates. But we must not be discouraged by that and persevere as well to get the traits that we want into the germplasm we need in those other latitudes. And of course, I say we can manipulate this with nitrogen, growth regulators, and if the variety has merit, we can do that to make it grow seed in the long day conditions of the summer for the winter season where there are short days in the latitudes, like I'm thinking of countries like Bangladesh. So now comes the next question. Why do European varieties and North American varieties do so well in the tropical zones? And this is just a picture showing arrows from Europe, from the Netherlands and UK and France and Germany, of varieties that have moved to different parts of the tropical area of the world, and they've done very, very well. Why is this? Here's a picture from Bangladesh. I used to live in Bangladesh and work in Bangladesh, and I used a lot of Dutch germplasm there. And here's a picture on the left of these women dehalming a crop of seed potatoes. This is a Dutch variety, very small canopy. The day length is probably 10 hours or 11. They tuberize early, mature early. Picture on the right, you see them harvesting the potatoes. Not a bumper crop, but they have a crop that they want to have because it fits into the cropping system. There they have a short growing season for potatoes in a rice-based cropping system, which is common in many parts of Asia. So very early tuber bulking is imperative. Small canopy is the result, but you'll have a crop at 60 to 80 days. And this could be improved a little bit by adding some more nitrogen. But here you see rice paddies all around this upper terrace is where there's still some potatoes that will be harvested shortly, and that'll also then go into rice. So under a system like that, these European varieties have done very, very well and have played a very important role in increasing food production in countries like Bangladesh and other Southeast Asian countries. India is kind of unique. They have made their own strategies and have developed varieties that are adapted to both the highlands and the lowlands as well, Kufri Jyoti being one of the old traditional varieties of India. And just to look at some of the most popular European and American varieties that have grown around the world, I just list a few here from the North America: Red Pontiac, Atlantic, Frito-Lay 1867 is a chipping variety, Red Lesotho, Irish Cobbler, Superior. I should mention the variety Shebedi. These have all come from North American breeding programs and have done very well for many, many years in more tropical settings. And then there's the European varieties that you see on the right, like granola, grown widely in Southeast Asia where there's more rainfall, like Indonesia, the Philippines, Sri Lanka. You have Mondial, you have Favorita, popular in China, Diamond, Mira, Mariella in Vietnam, Spunta in North Africa. These varieties are all traditionally grown for many, many years and are hard to beat because they are so well adapted to the short day growing conditions of these environments. However, these varieties all have deficiencies and are lacking in some abiotic or biotic stresses they can't handle. And that's why we're talking about what we're trying to do now with moving this work forward, what we're talking about here today. So, why do we need to further exploit the genetic diversity for the center of diversity? I think most of us are hearing more and more talk about sustainability, reduction in use of pesticides. There's a very strong push for this in Europe, here in North America, as well as in China and other countries of Asia. We must be more sustainable. And this, that requires less pesticide use. It means pesticides are being banned or restricted use. And hence we need more resistance to the stresses that the plants experience, and insects and diseases are primary. Then there's the issue of water supply. And I think it's very important to understand that drought tolerance and generally dry season potato production, water is a limiting factor. And if we can get better drought tolerance into our germplasm, into our varieties, we will again become more stewardly and more sustainable. Then there's the issue of late blight resistance, which still is our #1 issue around the world. How do we deal with this problem? And multigenic resistance is one way of coping with this. Then there's bacterial wilt, which is a disease in the low-level tropics, and it's becoming more severe. And as we have global warming, it seems to creep up into the higher elevations of the tropical settings. Of course, we all know about virus resistance, but you also think of processing qualities. And then we need to talk about biofortification with iron and zinc and other elements that need to be added, more vitamin C, vitamin A. These are all ways that we can improve the quality of our diets as well. And this is what's offered by the potatoes of South America, of the Andean region. So that needs to be further explored and utilized in our breeding programs around the world. So even as we think about the present and the future potato breeding strategies, a lot of work's being done now with cisgenics, with gene silencing, gene editing, gene mapping. We have a great efforts now on diploid breeding. And we also have a lot of traditional work continues on tetraploid crossing and selection work. But for all of these kinds of research and development work, and a lot of this is in the laboratory and in the greenhouse, we need to understand the practical application of how these things are influenced by the environment where we're trying to grow them. So day length and light intensity are two very important factors that play into this. That's why I wanted to share this with you today, because I think it's fundamental that we understand the environmental factors by geographic area as we move ahead with rapidly trying to improve potato production and become more sustainable in potato production around the world. With that, I want to end. If you want to have further reading, this is an article we wrote many years ago, but this article really clearly shows we used varieties like Red Pontiac and Desiree from Europe and North America, and we used varieties from SIP with diploid material in it and Foreca material, as well as Indigena. And we looked at different day lengths and light intensities in a replicated experiment over 3 years in the Philippines. And this paper is actually very interesting to understand the fundamentals of what happened with this kind of research. So if you're interested, you can look up this paper and read further on this topic from this work. With that, I want to thank you for your attention. And I think now we'll see how this works with our Q&A. So thank you very much.
So Peter, can you see any Q&A questions?
Yeah, so the first question is, hello, a greeting from China. And the second question is a question about, can I recommend some good books on potato breeding? And I guess what I should ask is for the person who asked the question to maybe email me at peter@sunrisepotato.com, and I can email him some references that he would maybe enjoy. Particularly, I think the book by Anton Havercourt that has just recently been published is a great book that we can utilize for this as well. And Anton has just done a beautiful compendium book That's a very broad book, actually, but it costs about $100, and you can buy it from Potatoes Europe. And I think this is what I would suggest. But anyway, if you email me, I will then go ahead and send you the details. That's great. I just put the website up on the screen. So there's a question here about true seed and about how to use true seed. Well, I think my talk today is really not focusing on how to use true seed, true potato seed, which for growing potatoes directly. I know that's a dream that we have for many parts of the world, that we don't no longer plant tuber seed, but move to planting true potato seed. And I think this is where is a challenge. And I think that we can do this, and I think work that's being done in numerous parts of in Europe, as well as in China and at SIP in Lima, developing true potato seed that is of larger size, vigorous, that you could sow directly and grow directly. Same thing still holds true as far as the influence of day length and light intensity on how the crop will respond. So no matter how you grow your crop, the environmental factors are going to play a big role in how the plant responds to the day length and light intensity. Okay, so the question here from Alberto Maurer from SIP. Can you please explain why long day potatoes can adapt to short days, but not vice versa? That's a very, very good question. Actually, it's something probably that I haven't really thought about well enough, that the genetic makeup of the potato is such that it can do the partitioning to tubers better under short days than under long days. I know of no variety that is day neutral at this point, although I think we can come to the point where we can find day neutral varieties. But this is just something that's innate in the potato, that this continues to be the case, that long days will produce, long day adaptive varieties will grow under short days, and the vice versa. I mentioned the variety Unica, or Chingsu 9, as being an exception. And that variety is, to me, is very interesting, because that allows us to explore that variety for that very important trait, to get more into having day neutral varieties. So I'm not sure if I answered the question well enough, but anyway, it's a start. So Dave Douch has asked, why can't we select potatoes in 2 environments? And well, that's exactly what my project is in Southwest China. I'm working on the agile potato, Dave, and the agile potato, we're working both in the summer highlands of Yunnan, and then in the lowlands of both Yunnan, as well as in countries like Bangladesh, Myanmar, Vietnam at this point, to see how we can grow them in both. And this is the challenge, and the work that you saw of Dr. Tung in Vietnam, he's doing the crossing work for that project, actually. So we've chosen a middle ground. So Dalat is at 1,300 to 1,500 meters above sea level, working in the highlands of Yunnan at 2,500 above, and then in the lowlands at close to sea level in countries like Bangladesh and Myanmar and Vietnam to do this work. And I think this is what end of the day, we need to have what I would call agile potatoes. And we're working towards that. Okay, the next question is about expectations on 2X breeding. Is it a game-changing technology? I think that this is definitely now very close to becoming a reality. When I see the work that's being done by Solenta in the Netherlands, both in Africa as well as in Europe, when I see the work that's being done in China, I'm very optimistic that because of the great knowledge we've accumulated over the years on the genetic makeup of potatoes, and the diploid is being so simple, we can make it homozygous parents very easily now, because we've overcome some of the infertility problems, and make these uniform progenies that are totally uniform. And we're going back to the maize of 1960, where the maize, the single cross maize was introduced, and the hybrid vigor was so great from the heterosis effect that I think we're in the same stage now. And I hope within 25 years, this will be a game changer in many parts of the tropics. Uh, there's a question from her way about how important is temperature in this whole complex of day length and light intensity. I would like to do a separate talk just on temperature, because temperature is also a very important aspect, and I think one that we have not overcome. Way, you know, our work in the Philippines, where we worked under very hot conditions, and there we found a lot of trouble with adaptation. Under short days, it worked fairly well, but even there, the gibberellins in the plant were very high. You get long stolons and very poor tuber growth and development. So this is definitely a challenge that we will overcome with time. We need to address this as we have climate change as well, and things are getting warmer. So there's a question here by Peter Joyce about getting sessile tubers on the tropics, opposite of long days, long stolons. I think this problem is partly to do with physiological age of the seed, if I'm not mistaken, if I understand the question correctly. And then because the age of the seed is physiologically old, the plants tend to tuberize too quickly. And because of that age problem. So physiological age is a very important factor in managing tuber seed. And this again is something that needs to be addressed, especially when you have to store seed for a long time from one rainy season to the next rainy season, like you do in parts of Africa. And same in Southeast Asia. That's why the agile potato going from the highlands to the lowlands allows you to have physiologically young seed. The challenges of breeding in sub-Saharan Africa. I think the biggest challenge in sub-Saharan Africa right now is bacterial wilt and late blight. Bacterial wilt is going to get worse and worse unless we have very strict sanitary conditions, but which farmers follow rotations, proper rotations, clean seed. But bacterial wilt is going to become more and more of an issue as potato production increases and temperature increases. Okay, the question here is about indigenous clones have distinct home characteristics that make it difficult to fit current agronomic practices. I think this question are these physiological traits linked to day length and adaptation to light and also light intensity? I can't answer that question, Dave. I'm not sure how that really fits together, but definitely the indigenous types have smaller leaves, smaller leaves, finer foliage, whereas the tuberosum generally have stronger stems, bigger leaves, and generally tend to be more adapted to adapted to the longer days. What's the key factors affecting the day length adaptability of the potato? I think the question here about adaptability of the potato is really a function of genetics. And as I said, if you look at Unica or Qingsu 9, compare that to, say, Cooperation 88 in China, they respond very different to long days, Unica being less sensitive, whereas the C88 is very sensitive. And so genetics is a very important factor in this, and I think we need to study that very carefully and utilize those that are less sensitive in further crossing work if they have other appropriate traits as well. Well, Mario Andrada asked a question about transferring genes from the wild germplasm to the elite germplasm without loss in yield. Well, I think this is where we have the work on cisgenics is the key. I see this as one of the great benefits now with the whole genomics work we've done, and then with cisgenics to move these genes from the wild species into the cultivated varieties is one of the very quick ways to improve existing varieties with the traits, whether they're biotic or abiotic traits that we want, as well as biofortification. So Dave Douch has asked a question: can we improve the work between Europe and US and the developing countries? You know, I failed to mention this in this talk. HSBC has done a great thing of working with SIP. They have a project which is actually supported by the Syngenta Foundation, where SIP and HSBC are now working together using SIP germplasm and HSBC germplasm, and working together to combine the good traits of South American germplasm into what HSBC has in Europe. And that is being evaluated in countries like Vietnam and Indonesia, and I think in Ethiopia. And I think more of this kind of linkage and collaborative work needs to be done. Michigan State could do the same thing. USDA could do the same thing too, where we have agreements where you can move true seed and pollen from South America easily to those other countries to utilize more effectively in creating more diversity in the gene pool. So I think HSBC's example is great, and we need to have more of that. I think I'm going to stop here. We've had a very good discussion. I appreciate all your questions. I'm not sure, Nora, if they can ask further questions that we can answer later, but I think our time has come to the limit. And I want to thank the great set of questions and discussion we've had with all of you. And this is the goal of what we have, is to link potato people from around the world together to address common issues and problems and to understand them better. So thank you all for your attention, and I appreciate this chance to share with you tonight and today. Thank you.